There we go. All right. Simple is good. Oops, I gotta grab my clicker. So you said not to start out your talk with but I guess you shouldn't start out your talk with oops either. So anyway. That's one of the fun things. You get to violate all the rules of speaking and still works. So as Michael was saying, the eKINDS project is a research initiative that was started by the Creation Research Society. Okay, let me spend just a couple moments talking about the Creation Research Society in case you're not familiar. Creation Research Society was founded in 1963, which makes us the oldest of all the major creation ministries. We predate all of them. You're aware of the museum and the Ark in Kentucky, right? Yes. Okay, that's not us. All right, that's Answers in Genesis. We predate them. Okay, you maybe have heard of the Institute for Creation Research that was founded by Henry Morris and Duane Gish. Okay, that's not us. Although they did help found the Creation Research Society a decade earlier, and that's what began to give Dr. Morris the idea that he wanted to actually establish an in-house institution, which is now known as the Institute for Creation Research. Okay, our website, I guess I didn't put the website on there. It's creationresearch.org, so it's not very difficult to find. And if you're not aware, we have recently moved our offices and our research facility. We used to be in Chino Valley, Arizona, and now we're on the campus of Arizona Christian University in Glendale, Arizona. And in so doing, one of our objectives was to just seek a little more high-profile location, seek a little more modern facility. We completely rebuilt where we are now. So it's a totally modern office space, a completely modern laboratory space, which we are working to equip. And the eKINDS project is one of the projects we're doing in that laboratory. All right, eKINDS. Well, there was publication of, and I'm going to have to make sure I remember what I was going to talk about in this. One of the purposes of eKINDS is to address the idea of rapid diversification. In other words, when all the animal pairs disembarked from the ark, they had to adapt to a completely new environment than what they had been in prior to that. The Apostle Peter writes about the earth that then was and the earth that now is. And one of the interpretations we would give to that, which is also then going to be supported by what we see geologically, is that the earth had totally changed. It had changed in its landscape. It had changed in its atmospheric conditions. It was not near as warm as it was. People talk about climate change, talk about global warming. We're still warming up and drying out after the flood. See, their models never account for that. That's why they're always going to be wrong, because they're imposing a fake history. And when you impose a fake history, you get a wrong understanding. You get a wrong answer, of course. Okay, so one of the purposes of the eKINDS project was to address, all right, the animals disembarked from the ark. They faced a completely new environment. How well did they do? And some of them, obviously, like dinosaurs, not very well. And they eventually died off. Some of them have done very well, but they're not necessarily in their original form they were as created kinds. And we're going to talk about that some. So here becomes the challenge then. The diversification that would have occurred, all right, how do we really look at this? How do we address this? All right, in the eKINDS project, we're going to say that with organisms today, they're descended from the same created kind. Which ones? Is this organism and this organism? Moose and elk come from the same created kind, for example. See, so that's some of the questions that we're working to address. All right? What mechanism is going to be involved in this diversification? And then can we have the natural history of various animals? Can we recreate that so that we can actually then understand how when the animals got off the ark, they repopulated the earth? Because that was one of the commands, of course, obviously, is populate the earth. So the historical context of the eKINDS project would be the biblical creation model, which says there's a creator, everything was created. In contrast to the evolution model, which says, of course, you weren't created. All right, the biblical creation model says that animals and plants were created according to their kind, and they reproduced according to their kind. Whereas the evolution model doesn't have those distinctions. We all really share a common evolutionary history. So we and elephants and alligators and fish and coral and sponges all share a common evolutionary history. Right? The biblical creation model says that everything was designed to reproduce and fill the earth. That was the intent God gave to it, and therefore he gave it the ability to reproduce and fill the earth. The evolutionary model says that only naturalistic processes can be involved, and this is basically depending on time, chance, and natural selection. Now, I'm not going to spend time really talking about evolution per se, but I just want you to understand the contrast of the two. All right, so this becomes then the question, and actually this is a question posed by the website BioLogos, which, if you're not familiar with BioLogos, it is an evolution website that poses, and I'll be very blunt, they pose as Christians. Saying, okay, see, here's what, quote, science says, and therefore, as Christians, we need to work this, incorporate this into the Bible. So one of the questions they ask is, does modern animals evolve from the inhabitants of the ark? And of course, we would say yes. They say no, we would say yes. Our challenge is to answer, okay, you have the original created kinds with some diversification that occurred. We probably would say not this much that they show, but some diversification occurred. Then you had the flood, and then since the flood, there's been a much greater level of diversification, getting us to the level we see today. All the diversity of plants and animals we see today would have stemmed from those animals that were on the ark or those marine animals that survived the flood. So, this brings us to a good starting point in that respect, and that is to understand the difference between kind and species, because we're talking about that, that God created kinds. And many people get this confused, say, well, he created species. Well, no, he didn't. Kinds and species are not the same thing. What is a species? It's intended to represent the lowest level of biological diversity or classification today. But that would not reflect what it was during the creation week. That level of diversification or lowest level of classification would be much different the creation week than it would be today. So already we understand that kinds and species are not the same thing. So we got to recognize when the Bible talks about kinds, that doesn't mean species. Species is a man-made concept. And as such, that means it's already confusing, because humans are always confusing when they're trying to do stuff. Okay, one of the primary ways of defining a species is the ability to interbreed. Now, problem number one, that already excludes all of the asexual organisms out there. And if you're going to include the microbial world, which is obviously in biology sense, very much part of biology, then that excludes some 90% of all biology, because 90% of all biology is asexual. Now, if you're going to focus just on plants and animals, see, then it's got to be those plants and animals that engage in sexual reproduction, the ability to interbreed. But here's a problem. That's not always very clear, especially in plants and in birds and in some forms of insects. You'll have organism A can interbreed with organism B. Organism B can interbreed with organism C. So it would seem that A, B, and C are all the same species. except A and C can't interbreed. So where's the species line? No one knows. It depends on if you're what they call a lumper or a splitter. See, if you're a lumper, you'll say, well, A, B, and C are still all the same anyway. If you're a splitter, you're going to make A, B, and C all three different species. See, there's some 1,200 species of fruit flies in the Hawaiian Islands. Are there really? No, of course not. But you don't publish a paper talking about what you discovered that someone else has already discovered. You publish a paper talking about a new species. So you have 1,200 different species identified to give you 1,200 different papers. But the problem is that gives the impression of all this diversity that really isn't there. You really don't have that level of diversity of fruit flies on the Hawaiian Islands. All right? Also, geographical distance can create a species distinction. We'll talk about this a little bit more in a second. But you can have organism A and organism B can interbreed, but organism A lives in Africa and organism B maybe lives in Asia, and they never get together. Therefore, they don't naturally interbreed, and so the biologist says, well, they're not the same species, even though they can interbreed. See, so that adds also to the confusion of, well, what's a species? Okay? And how do you apply this to a fossil? For all my years, I've never seen two fossils interbreed. They just don't do it. So you have a problem. How do you know what it interbreeded with? Interbreeded, new word, yeah. How do you know what it interbred with? You simply don't know. Here's an interesting quote. How many of you have heard of the fossil Lucy? All right. Donald Johanson was one of the original discoverers and curators of the fossil Lucy. And in his book about Lucy, The Beginnings of Mankind, he makes this comment. He said it would be interesting to know if a modern man and a million-year-old Homo erectus woman could together produce fertile children. The strong hunch is that they could. But notice it's two different species. We're Homo sapiens, they're Homo erectus. He says, but this does not violate the law of species definition of interbreedability. No, it doesn't violate that. Even though you're saying they could interbreed, but they're two different species. No, we're not violating that because they can't interbreed because they're separated by time. Now think about that a second. They're separated by time. If they were together, they could interbreed. So biologically, they're really the same species. But they're separated in time, therefore they're not the same species. Okay, so does 19th century man and 21st century man, two different species? Don't answer that, but anyway. But you see the point. The point is when you start imposing then that kind of arbitrary distinction in the species, you create even more species than there actually are. So what I'm trying to get at is that, yes, we as creationists have to account for a certain level of diversity that's occurred since the flood, but probably not as much as people think because they've imposed all this diversity of species that probably doesn't really exist. And therefore, there's not nearly as much diversity as you think they are. See, people talk about, well, there wasn't enough room on the ark for all the species. No, you're right. Probably not. But it wasn't species. It was kinds on the ark. Because the species today didn't exist back then. Especially when we talk about these arbitrary species that they just made up because that's what they want. They want this diversity. And let's face it, the evolution loves speciation because they think that's what drives their theory, even though speciation actually is the opposite of what they want. But they like lots of species, so they think that makes evolution true somehow. All right, genetic analysis has helped then to clarify some of the species boundaries, but not all the species boundaries, because what's your criteria? How close does it have to be the same species? 1%, or excuse me, 99%, 99.9%, 95%? See, some species, they're 99% similar and they're still different species. Other species, they're only 90% similar and they're still the same species. See, again, there's a lot of arbitrariness even when you're using genetics. So it's still very confusing. Even to biologists, it's very confusing. And to Bruce Jenner's dog, it's very confusing. See, so the confusion just reigns when you're talking about human thinking, human distinction, human definition, be it human definition of species or human definition of male, female, okay, it gets very confusing because we humans, we're just confused. That's all it is. We're just simply confused. All right, so back to what I mentioned before. What's a fossil species? How do you know when you find this bone here, it is or is not the same species as the bone you found over there? All right, if we only knew about these two creatures based on their skulls as fossilized structures, would we say they're the same species or not? My guess would be we'd say they're not the same species. Just based upon you have a different cranial structure. This is more round, this is more rigid. You have a different eye socket structure. You have a much wider jowl structure. You have a very pointed one here. The teeth structure is different. The nose structure is different. So yeah, it must be a different species, right? Okay. Look at all the diversity we see just within dog breeds, which, by the way, dogs would be different species if we didn't know they're all the same species, so we call them breeds instead. But believe me, there's more diversity between one dog breed and another there is between those fruit flies in Hawaii. Okay, so the only reason we don't call all these different species is because we know they're not, because we know the history of them. If we didn't know the history of them, we'd probably call them all different species. But see the diversity that occurred just within dogs. Look at this. What would be the paleontological interpretation if you saw this in the fossil record? You say, wow, look how we've had evolution of the skull over millions of years. Yeah, less than 50. Less than 50. And what is pointed to is there are events going on at the cellular level, at the genetic level, that we're only beginning to understand that can bring about massive transformation of certain features, like the skull structure of the purebred bull terrier. But how would paleontologists interpret this? Well, I think we already know. If all they knew was just these skulls, this would be an evolutionary lineage over millions of years, even though clearly that's a wrong interpretation. What we now understand is that within your DNA, and if you understand within DNA, we write out DNA sequences based upon the sequence of what's called the nitrogenous bases: adenine, thymine, cytosine, guanine. The backbone of DNA never changes. It's the same whether it's bacteria, whether it's a virus, whether it's a human. The backbone is always the same. What changes is the sequence of the adenine, the thymine, the guanine, and the cytosine. So when we write down a sequence, what we're really doing is just writing down the sequence of those 4 bases. What we know now is that you have repeated sequences. Notice this sequence and this sequence is the same, and then we repeat it. And then we repeat it again in another organism. Repeat it again. When we repeat those sequences, the more repeats, the more morphological change we introduce, just by repeating those sequences. Also, there is an 18 to 20 codon. That's a certain 3-base sequence that is for the amino acid glutamine and is followed by 12 to 17 codons for alanine. In other words, you have 18 to 20 of this one type of codon, you have 12 to 17 of this other type of codon, and if they occur in tandem in the DNA, that gives you a certain morphological feature. You change the number of those repeated codons, you change then the morphological feature. And it's these two together that they are surmising is what led to this. Just that, just those repeated changes in the DNA gave you this kind of change in less than 50 years. See, so we can understand how rapidly, once the animals got off the ark, that they could have begun to change physically so that they would not necessarily look like those animals that came on the ark a few generations earlier. In fact, I would go as far as to say that if you were to transport yourself back in time, this is what AzOSA is trying to do with their ark. It's an interesting challenge for them. If you transport yourself back in time and looked at the animals that Noah had on the ark, many of them you wouldn't recognize. What is that? What is that? Because what you see today are its diversified offspring, and they have morphologically changed because there's now been introduction of repeats in their DNA. There's been other events we're going to talk about in their DNA that have caused changes to the skull, caused changes to the body cavity, that have led to changes such that they don't necessarily look like they did when they were on the ark. Okay, so let's go back to this guy. Is this same species or not? Anybody know what these are? Nope. Bigger than that.
One's a wolf.
Not a wolf. You're getting close. You're getting close. You're getting close. St. Bernard and a Chihuahua. Now, no one would tell you that these are different species because we know they're not. And yet, if all we knew about them was some fossils in the fossil record, we almost certainly would say they're different species. So you begin to see how it's very easy, based on an evolutionary interpretation, to misinterpret the fossil record. And I say that's what we're doing all the time. They're misinterpreting the fossil record because, first off, change can occur rapidly, but it's a limited change. There's still a dog. Okay? But you can see tremendous diversity within the dog kind, but it's still a dog. It's still part of the dog kind. Okay? Variation of cattle breeds. Again, the same thing. These would be different species if we didn't know the history of them and know that they're really the same species, just different breeds. Where do you get a miniature horse from?
Big horses.
Yes, from big horses, exactly. Very good. What you do is you take a big horse, you breed it, and you take the smallest horse that you find from the offspring of the big horse, and you breed it. And you keep doing that. You keep pulling out the smallest and the smallest and the smallest, bringing out a miniature horse. The miniature horse is not a separate species. It's a big horse that has had all of the genes for big horse pulled out of it. And now it's small. But it's still the same species. But again, demonstration of the tremendous diversity that can occur within the created kinds. And once again, because we know where this came from, we know the history of the miniature horse, we recognize it's not a different species. But the challenge would be, if all we knew about it was from the fossil record, would we classify it as a different species? Almost certainly. All right, cat kind. Creationists think, and you got to recognize we're trying to do historical reconstruction, and anytime you do historical reconstruction, it's only worth, so much. All right, but we think, based on what we know anatomically, based on what we know genetically, we think that whatever the original cat kind looked like, and this is just a very, this is just there. Wouldn't even begin to say it looked like that. Okay, it obviously had a tail and 4 legs, but more than that, we really can't be certain. But from that original cat kind that got off the ark, we now have lions and tigers and jaguars and bobcats and house cats. So your house cat is actually part of the same original created kind as tigers and lions. You can cross a lion and a tiger. Were you aware of that? See, lions and tigers are geographically separated. So they're considered different species because they're geographically separated, but they can be crossed. If you cross a male lion and a female tiger, you get what's called a liger. The liger is bigger than both the tiger and the lion.
I have a friend who has a zonkey.
Yeah, we're getting to zonkies. Yes, yes.
I didn't think really
Yep,
were that popular.
you are ahead of me. Yes, yeah. The liger stands about that tall. They're massive creatures, and that begins to tell you probably what the original created kind cat was probably a huge creature. Okay, if we do a male tiger and a female lion, we get a tigon. They're not as big, and interestingly enough, they have kind of more the striping that the liger didn't have.
And Kevin,
Yes.
the offspring are also fertile and continue to reproduce.
Some of them, yes, yeah, can be, yep. The domestic cat and the serval give you the savanna cat. Okay, Now, not to mislead, but if you take like a bobcat and a house cat, they can reproduce, but it'll be sterile. So sometimes some of these interbreedings will give you organisms, give you offspring that can reproduce, but other crosses don't. But that all fits within the created kinds model because it all depends upon what's been lost. We would say, see, tigers and lions are enough genetically similar. There hasn't been that much lost through the diversification. Go back to what gave you this miniature horse. The loss of the features that gave you a tall horse. What gives you a domestic cat? Losing the genes that give you a lion and a tiger. And the domestic cat's lost enough that it doesn't interbreed with giving you reproducible or children that reproduce. Yeah, it doesn't give you a offspring that can reproduce, but it can interbreed with other types of cats, like bobcats, for example. Right? Bear kind, we see the tremendous diversity of bears. Most bears can interbreed. In fact, you know the grizzly, you know the polar bear, okay? So have you heard of the prizzly? In fact, I think, do I have a, yeah. Here we go, yeah. The polar bear and the grizzly, see, they can interbreed. Grizzlies and black bears can interbreed. See, so they can interbreed, they're just separated geographically, so they're identified as different species. And notice, see, what's happened with the diversity that's occurred. The polar bear lives where? He lives up in the northern regions where it's much more snowy, so the white serves him for an adaptive purpose. The grizzly bear and the black bear live within vegetation, so their black and brown coats serve a better adaptive function where they live. But see, that's all part of the diversity that God's programmed into these animals to allow them to get off the ark and fill the earth, even though the earth has a variety of different ecological niches. Okay, we go to our dog kind, which we've already seen, but see, of the dog kind, we think the dogs actually originally came from a type of wolf. So we think that the original created kind that Adam would have played with and Adam's sons would have played with would have been more what we would call a wolf. And from that, we've now had dogs, coyotes, jackals, and perhaps even foxes. Fox is a little uncertain, but we think so. Okay. Here you have the various types of horses that would have diversified from the original created kind of horse. See, so here's your zonkey. Okay, a donkey plus a zebra. Don't you love the socks on that thing? See, but that again gives us an idea of these would have been offspring of, they are part of the diversity from the original created kind. Whatever that original created kind horse looked like, we're not totally certain, but from it would have arisen the horse as we know it, the zebra, and the donkey. These would all be direct descendants of that original horse that God created and then was also on the ark. And then you have a horse and a zebra, you get a zorse. Much more striped. Interestingly enough, genetics actually helped work this out some. We kind of assumed that porpoises and dolphins probably were fairly genetically similar, so that's not a surprise. So they would have come from the same original created kind. But we also now know that killer whales, orcas, are porpoises. So they would have come from the original created kind, whatever again that looked like, we don't know exactly. So once more we see the diversity that would have occurred since the flood, or really since creation in that regard. And this doesn't really speak to any of the diversity, it's just I love this picture. Because to me this kind of hints at maybe what it was in the very beginning and what it will be again. And reading, I've had this picture for years, but if you read the story, what happened is the bear and the dog grew up together. And the dog is the bear's best friend. The bear loved to go hug it all the time. And I just think that's so cool, because that's the way I think God originally intended it to be. All right, identifying created kinds. Well, how do we know what the created kinds were? Well, we don't totally. We went through a little bit of how we can go about it. When they were trying to do the ARC encounter, they had to pose the question of, okay, what would the original created kinds have been and what would they look like? Well, we'll make some guesses. Okay? Hybridization data was used. In other words, how similar are the DNA sequences within different organisms? The more similar, the more you would think or conclude that they might have been from the same created kind. All right. Also, then, what's called Cognitum ontology data. In other words, this is a specific analytical analysis of data that allows us to take the physical features and compare them to see similarities. Okay, statistical baraminology data, and that's really kind of involving a wide process of information. Barams is often what's referred to as the created kinds. We're going to divide this into barams, which would be analogous to the created kinds. Okay, also then, some of the limitations, of course, we don't have hybridization data for all organisms, so obviously you can't do that. We don't have statistical analysis data for everything, and there's not enough data to do all the statistical analysis. So there's lacking data yet to be able to do some of the comparisons we want to do. So we'll get better at defining what the original created kind might have been as we collect more data and get a bigger pool of genetics, for example, and anatomy and proteins and such. Okay, some of the results were that it has been estimated, and most biblical creation geneticists agree with this. In fact, I think they probably all do. Can't think of any actual trained creationist that's a geneticist that would disagree, that probably in our current classification scheme, if you remember from biology, you have domains and kingdoms and phylums and orders and classes and families and genuses and species. Probably the original created kind would fit more at what we today would identify as the family level. And you diversified into genuses and species from that. Because when you get past the family level, then all of a sudden it breaks down as far as trying to make links. See, lions and tigers and your house cat are all the same family. That's the feline family. Go back past the family, all of a sudden now there's not a good linkage anymore. And I'd say that's because it doesn't exist. Because the family approximately is where the created kind was. See, evolutionists want to go back and link then the classes and the order and the phylums together as well. But see, the data isn't nearly as good because, in my opinion, it doesn't really exist. Because you didn't have something that evolved into a cat. You didn't have something that evolved into a dog. You didn't have something that evolved into a horse. Horses have always been horses, even if they don't look exactly like they do today. Cows have always been cows. Dogs have always been dogs. Cats have always been cats. Even if physically they're a little bit different than what they were always a cat. They weren't something evolving into a cat. All right. This is a paper that was published in the Creation Research Society Quarterly a few years ago. And this is a PID name, because at the time that the scientist wrote this, he was still in school and he didn't want it getting out because, unfortunately, you recognize that if you're a creationist, you're going to likely get blackballed and ostracized and all of that. Because, of course, you know, the secular scientific world is so open-minded and supporting of free speech and all of that stuff, right? Yeah. Yeah, and there's a bridge over there I'll sell to you also. Okay, anyway. But what he did was he took what's called the proteome. Okay, you've maybe heard of the genome. The genome is the collection of all the DNA, all the genetic information on the DNA within an organism. The proteome is all the proteins within an organism. And he took the data of all the proteomes of different organisms, compared them. So he's comparing protein sequences instead of DNA sequences. And what he found was, which I find particularly interesting, is that here is human, and here is primate, and they do not overlap. Very distinct difference in human and primate. Not what the evolution community would have you believe, because they want you to think that, you know, chimpanzees are almost human. You know, they're so close, they're almost human. Okay, well, you can have a chimpanzee live in your house for a year and see how much you think they're human, but that's a whole other story. All right, but what he found based on this proteome analysis is that our protein collection in our bodies and the collection of proteins in the primates are very different. Therefore, you really don't have a basis saying that we and chimpanzees and orangutans and such are all that evolutionarily similar, because we're not. There's a great distinction between us at the protein level. Now, one of the problems of this approach, it's a very good approach. One of the limitations of it is you have to have the complete proteome of an organism. And by and large, we kind of lack that for a lot of them. Now, that's going to change over time, but we still don't have enough data yet of enough organisms to be able to do too much of this type of comparison. Okay. Also, evolution needs massive gene changes to explain the origin of orders such as in the insects, where changes within the orders can be explained by gene loss. In other words, we can explain to you from a biblical creation perspective why you have such diversification in the insect world by the removal of genes, where evolution would say somehow those genes had to get put in. And we're saying, well, God put them in, but processes post-fall, post-Genesis 3, are now taking them back out, and that's part of what leads to speciation. That's part of what leads to diversity. Now, specifically know that for the insects, but it's not just the insects. We're finding out more that even within the mammal world, that it's diversification because of loss of genetic information, genetic structures, genetic sequences, genetic features, genetic systems. You lose those, and that creates then physical differences. That creates anatomical differences. And as such, then, that's part of what contributes to all the diversity we see today. And then, of course, humans are fully distinct from primates, is what he found, and other factors that might influence the groupings, such as the sample size and the density. See, and that is a bit of a limitation. We need more data to have a bigger sample size so that we can have a cleaner analysis. All right, mechanisms. Well, first mechanism is created diversity. This is one of the things that always amuses me, because when we're talking to our critics, they seem to never want to think that God was capable of diversity. They almost want to make Adam and Eve clones. Well, you may be confused on the difference between male and female, but God isn't. Okay? So they're not clones of each other. We know already they had some genetic differences. But even within Adam himself, he probably is what we call heterozygous, which means he had two different sets of alleles. That would be created diversity. And in that respect, then, their children would automatically be different than them, just as your children are different from you. But probably even more so. And I'll show you a little illustration of that a little later. But probably even more so because they would have greater diversity than we have today. There was probably more genetic diversity between Adam and Eve than there is between you and your spouse today. Because God would have created it. Because that would be part of their ability of adapting and living in an ever-changing world. So the idea that evolutionists always want to seem to act like that everything is supposed to be a clone. If it's created, they shall be clones of each other. That's just imposing their view in order for them to be able to criticize them. It's their straw man. God would have created diversity into animal populations, into plant populations, so they could reproduce and fill the earth. All right? Also then, DNA, what's called DNA editing. This is where, for example, you have what's called crossover, where alleles will move from one gene, one chromosome to another chromosome. Or you can have what's called gene conversion. Those are all processes that occur at the DNA level that can bring about variation, can bring about some levels of physical diversity. You would have possible causes of adaptive mutations. This would be a mutation that, for example, how many of you are not allergic to milk? Yeah, most of you, which is not surprising, because most of you probably have ancestry that existed at some point in time in Northern Europe. There was a mutation about 2,000+ years ago in the population in Northern Europe that allowed them to not have a problem digesting the milk, sugar, lactose. See, 70% of the world's still allergic, if you will, allergic. They don't digest lactose very well. I'm glad I have that mutation myself because I like milk. I like ice cream, you know. So if I didn't have that mutation, then I'd be in trouble. I wouldn't be able to necessarily consume what I consume. So for me, that's a nice mutation to have, but it is a mutation. But it introduced variation. It introduced a type of diversity. It introduced an adaptation. Because the people in the Northern Europe, they were relying a lot on not crops so much as milk from cows. And so that was an adaptive ability that they had because of that mutation. Okay, adapt for specialized environments, just as that. The darker skin, typically where those people, they're in more of the tropical regions where the sun is more intense. So that was an adaptive mutation that allowed them to be in a more intense sun environment. Our white skin is really the last of the pigment mutations to occur in human history. So we're really the latecomers, despite what some white supremacists want to say. We're the latecomers, okay? But again, Northern Europe didn't have near the sun intensity. And so what you found was the light skin actually functioned better for them because they didn't have to worry about the problem of all the ultraviolet light if they lived in a more tropical region. All right, mutations then, we mentioned mutations here, mutations that can be random and non-random. This is driving the evolutionist community crazy because it used to be the assumption all mutations, you know, a mutation is just simply a change of that adenine or that thymine or that cytosine or that guanine. It should be an A, but a mutation makes it a C. It should be a T, but a mutation makes it a G. It's like just a change one letter on the page of your book. That'd be a mutation. It used to be assumed that all that was just random. So you never knew which letter was going to change. We now recognize there's many mutations that are not random, and that's a whole nother talk, maybe next year. But we now recognize that not all mutations are random. Okay, explain how you got a process of non-random mutations by mutation. So you can't do it. You can't do it. So the fact that there's non-random mutations is driving the evolutionist community crazy because that doesn't fit what they have to have for their mechanism. But yet, see, for us, that's perfect because God just programmed in the ability of these mutations occurring in a non-random fashion. You needed that change. You needed that T to become a C in order for a certain pigment to be made or a certain pigment not to be made. It's programmed in there. There's some bacteria that intentionally can flip their DNA under certain conditions. And in so doing, then they can adapt to that condition. For all we know, we can do that too. We just haven't figured that out yet. Okay, number 4, hybridization. In other words, you can take DNA and bring it together, and if it's similar enough, it will actually fuse together. And there are organisms where this does occur. And so there's been some changes that occur because of this. Now, again, as biblical creationists, we say that there's a certain limitation that occurs. Evolutionists like to say, oh, this can explain all kinds of, this can explain where the fish became alligators. No, no, no. Not nearly like that. You don't have anything to back that up. But we do see hybridization can explain some levels of change that occur. All right? Horizontal gene transfer. In other words, I have a piece of gene and I'll give it to you. This occurs a lot in the microbial world. It occurs some in plants. It doesn't occur very much in the higher animal world. But evolutionists say, well, it occurred a lot in the past, not so much today. Okay? Natural selection. Again, that's a whole different subject as far as everything to be said about that. But even as creationists, we recognize that if you are in an environment where you are benefited to have the mutation for white skin or to have the mutation for being able to digest milk, then that mutation will probably become more easily fixed in the population because that provides you a benefit of survival. So even creationists understand that. Our difference is that natural selection is not what caused the mutation. Natural selection is what says that mutation actually is now beneficial for you. But it's God's programming that caused the mutation to begin with. Okay? And then we would say that, let's see what time, okay, non-Mendelian inheritance. Oh, genetic drift. I'm sorry, genetic drift. This is where you separate populations. And when you separate populations, then they don't have the same gene pools. Example, Tower of Babel. You've heard of the story of the Tower of Babel, right? this family group goes this way and this family group goes this way, and genetic drift would have began to occur, because if this family group and this family group are not interbreeding with each other, then the gene pool for this family group is going to remain isolated from this family group, and therefore you don't have a remixing. And this is one of the many things that would have led to the diversity within humans, for example. But you know, it wasn't just humans, they would have taken their animals with them. Do you ever think about that? We would also have genetic drift of the livestock and the pets. And that's why you find certain animals in some regions and not in others, because that's what they would have taken with them. All right? Non-Mendelian inheritance. You remember Mendel and his laws of inheritance, and all that's true, but we now know that there's also what we call non-Mendelian inheritance. In other words, changes that occur that don't fit within Mendel's predictions or Mendel's studies. For example, what we call meiotic drive. This is based on a process called gene conversion. This is simply, again, something that occurs at the DNA level itself prior to reproduction, but yet it introduces variety into the gametes of the parents. Okay? This can cause preferred transmission of specific alleles between generations. See, Mendelian inheritance says there is no preferred selection of alleles when it comes to forming the gametes. That's the law of segregation. The alleles are going to separate randomly. But we understand that because of meiotic drive, you can actually have some alleles that are not going to randomly be separated. They're going to stay together. And this kind of blows Mendelian genetics as far as doesn't fit his predictions anymore. And that can introduce diversity. Okay? Prevalence of alleles. Do you have a lot of these alleles? Not many of these alleles. This can affect your diversity. This can affect your physical characteristics. It can affect how rapidly the changes occur. Again, genetic drift. The founder effect. Founder effect is a type of genetic drift. It's called founder effect because it's based on the idea that if you have a small population that isolates itself from everybody else, then that gene pool becomes its own unique population with its own unique physical features, often to the detriment of the population if it's small enough. But this introduces the idea that you can have changes occurring just simply by isolation. And it'll say here, see a lot of people that got off the ark, or excuse me, that would have separated after the Tower of Babel would have engaged in founder effect. So we would have seen then this level of diversity beginning to occur because of the isolation and the separation of the gene pools. And this is one of the factors that would have led to what we today call races. Even though geneticists for decades have been telling society there's genetically no such thing as race. It's just a matter of prevalence of alleles and the expression of those alleles. We all have the same pigment genes. It's just some of us don't express it as much as others because we have mutations that block the expression, where people that are darker than us express more of those pigment genes. That's what, but we still have the same pigment genes. So there's no genetic basis for race, especially based upon color, especially based upon hair texture. There's no genetic basis for that. It's physical. It's what we see in humans. Let's face it. Tell me the physical difference between Irish and English. Yet they hate each other. See, because we humans will find something to hate each other about. This is what sinful man does. So if you see someone that doesn't look like you, then it's real easy to hate them. Well, they're different. And so that's where the whole thing originated. As creationists, we should never have any kind of prejudice like that because we all recognize we came from where? We came from the same parents. See, evolution doesn't teach that. Evolution doesn't teach that. It teaches actually you did have diversity. Darwin thought that certain species, certain races were more superior than other races. We don't have to understand what he thought was superior and the inferior. Same thing Hitler did, okay? But as creationists, we recognize that's not true. We're all the same descendants of Adam and Eve. Okay, and then you can introduce adaptive alleles into the population through various types of mutations, various types of hybridizations, etc. that we've been talking about. All right, here's a fascinating one that creationists ought to exploit a lot more than they have to introduce diversity or variation into the biological world. And this is called phenotypic plasticity. Ironically, this is not based upon changing the DNA sequence nearly as much as it is simply taking what's already programmed into the DNA sequence. But what it's saying is you can have rapid morphological changes, but they're limited. Again, have to emphasize that. They're limited. This is not a horse evolving into a kangaroo or anything like that. This is still a horse, still a dog, still a cat, still a human, but you can have tremendous limited morphological changes occur within a small, that only involve small genetic changes. Yet paleontologists really on the average don't account for this when they interpret the fossil record, hence back to the Chihuahua skull and the St. Bernard skull. They don't account for what's called phenotypic plasticity. Okay, a few examples. Many marine creatures can change their size and shape, especially the crustaceous, which, by the way, what is 95% of the fossil record? Yeah, crustaceous marine creatures. So most of what we study fossil-wise are these guys, okay? We know that they can change based upon the presence of predators, nutrients, and other environmental factors. Well, how do you know when you find this fossil what its environmental factor was? Was there predators around? Was there enough food around? How do you know? The answer is you don't know. You simply don't know. So you don't know what kind of phenotypic plasticity was introduced into the population at any given time. Okay? I would challenge most changes we find in the fossil record from kind to kind, all right, are going to be because of rapid genetic change, response to stress, response to food availability, and response to environment. In other words, because of phenotypic plasticity. Temperature change will cause clams, for example, to change their size and shape. So because of that, we can find this in the fossil record. Now, is this an evolutionary progression, or is this just the same species under different environmental conditions? I would challenge it's the same species under different environmental conditions. But because evolutionists want to, they have to find their transitions. They would love to point to this and see the evolution of the clam. No, not anything of the kind. Phenotypic plasticity explains this very well. Presence of predators, guppies in particular, respond to the presence of predators where you will have them change their shape and size based upon their predators. If there's a predator, they get smaller. No big surprise. So they can hide easier. Okay, so again, when we're looking at the change occurring, this occurs very quickly. This is like a million-year process, or even a 100-year process. This occurs in the next generation. They're smaller. And in fact, we're going to go into that in just a second in some things. Okay, 2-year study observed changes in guppies equivalent to 2 million year change with standard interpretation of the fossil record. See how they're misinterpreting the fossil record, imposing diversity that's not really there in an evolutionary interpretation, which fits really much better the biblical record and helps us understand not only diversity we had at the time of the flood, but then diversity we've had since the flood. Epigenetics, and this again is a whole nother talk, but epigenetics is events that occur at the genetic level that do not involve changing the sequence of the DNA. Batteries in these things. Is that back? Is it still there now? Okay. All right. I guess wiggled around that, moved the battery some. Okay. Epigenetics is like not changing the letters on the page, but putting little marks, kind of like what the ancient Hebrew used to do, little dots and tittles, marks that change the word without changing the letters. That's what epigenetics does. You don't change the A's and the T's and C's and G's, but you change how the A's and the T's and the C's and the G's are read. So here's some mechanisms of epigenetics. You got DNA methylation, you got chromosome structure, chromatin remodeling, you got histone modification, you have the acetylation of the DNA and of the histones and such. All these are markers and tags to change the shape, to change the orientation of the DNA in the chromosome. And here's a consequence. Now, this is behavior, but it gives you an idea of what can happen based on epigenetics. If we take Mickey Mouse and we expose him to the smell of cherries, mice love cherries. Expose him to the smell of cherries, but along with that, we give him a mild shock, which he doesn't really like. Whoops, come on. So that agitates him. And we keep doing that. See, we're then going to have a conditioned response so that even just the smell of cherries without the electric shock causes an agitated response. Now, you've seen this before. Pavlov's dog. Okay, you condition them, they respond even without the shock, even without the stimulus. All they have to do is just get the cherry or get the aroma, whatever it is they're conditioned to, and they respond as if they're going to get the negative reinforcement. So you've seen this before. But here's what Pavlov didn't do. The offspring of those mice, when they smell cherries, they still react agitated, even though they've never been shocked. They've inherited the conditioned response of their parents. It's not because of the DNA, it's because of the epigenetics. You can inherit epigenetic tags of your parents and even your grandparents. If grandma's a smoker, it often will affect granddaughter. Remind a little bit where the Bible says that the sins of the fathers visited upon the sons into the third and fourth generation. All right? Now remember, we were talking about phenotypic plasticity. Predators present, so the guppies got smaller. Okay, you can take a small rump mouse and put it in a pen with a large aggressive mouse, so that's traumatized. Upon reproduction by a natural process, its offspring are runt traumatized mice. But if we do the same thing, but it's artificial insemination, so the female never sees the coward little runt mouse that's the father, you get normal sized, normal behaving mice. We don't know how. We don't know what's going on here. But this is epigenetics affecting what we would call phenotypic plasticity. These are examples that we can point to show how the animals that got off of the ark were able to diversify within just a few thousand years to what we see today. Even though we're often criticized, well, you believe in evolution more than I do. No, I don't. We just believe in rapid diversification, as exemplified by all of this I've just shown you. We certainly don't believe that some creature evolved into a horse, which evolved into a cow, which evolved into a dog. I'm not saying that's what evolution believes. I'm just using that as an example. Or the fish evolved into reptiles, which evolved into mammals, which evolved into humans. We're not saying that. We don't agree with that. But we are saying that the created kinds have diversified. That the house cat and the leopard and the lion and the jaguar and the tiger all came from the same original created kind. We are saying that, yes, based upon these mechanisms we've just shown, plus maybe some we haven't discovered yet. Okay, finish with a couple things. These are twins. Now, I'm not saying Adam and Eve would have had one dark and one white, because white didn't exist back then. Okay, Adam and Eve would have been substantially darker than we are. But I am saying that as we begin then to remix the human pool population, so we're no longer isolated groups, you're going to get more and more of this, showing the type of physical diversity that can occur within the human population. And we're actually the least diverse. Humans are the least diversified of all of biology, plant and animal and microbial. We're the least diversified. And yet we can still get this level of diversification. I'm sure they drive their teachers nuts. Your twins? Your twins? Oh, you're lying to me because we don't think in those terms.
Lucy and Marie.
These are clones. Now you understand, clone, they're identical DNA. But they're different. Exactly. This is epigenetics. This is epigenetics. How it affects where they're the same DNA, but they're different. This is a single generation. It didn't take millions of years. Single generation. So again, I'm hoping in all of this, and I know I kind of blew a lot at you, but I'm hoping in all this I can give you just a little flavor of why we can expect from a biblical creation model within a short period of just a few thousand years, we can get all the diversity we see today. It doesn't require the evolutionary interpretations that they're going to claim it requires. And so when you see in National Geographic or you see someone else who's trying to argue that creationists can't explain this, yes, we can. You're just not paying any attention. We can explain it much better than you can, in fact. So with that, just close with that our goals of the project, come back to the eKINDS project again. Our goals of the project are first to honor God by studying His creation and discovering the truth that that reveal His glory. Because remember, what does Paul say in the first chapter of Romans? The creation reveals the glory of God. The whole reason He created was to reveal His glory. And if we don't understand His glory revealed in creation, we don't understand creation. All right, to understand the mechanisms of diversification and explain how these point to our Creator, created complexity, He designed us for change, and He designed mechanisms that allow rapid change. It doesn't do an organism any good to go into an area that's much, to enter into Arizona for the first time. If you can't adapt to the Arizona climate quickly, you're going to be dead. Won't do you any good. So you got to be able to adapt quickly. And God programmed these, as you just saw, these quick adaptations that can occur. So with that, I will close a few. If you're interested, we have a book table here, as Michael's pointed out. We have our newsletter for free. You can sign up for if you haven't already. It tells you what's going on. It tells you about the eKINDS project, for example. And some resources, if you're interested, that you might want to pick up just to talk more about some of what we're talking about tonight, as well as other things involving genetics and involving diversity and involving just the creation model. So with that, do we have any questions? Went a little longer than I had thought. I apologize for that.
No, you're good. Speaking for myself, this was awesome. Yeah. And the monkeys applauding, I think we can see.
My wife always says it's self